Alright guys, back with you here for part 2 of chapter 21. In the previous video we had discussed the ability of cells to become determined to have a specific job, and then ultimately those cells became differentiated to actually do that particular job, as in the case of the myoblast cell that was determined to become a muscle cell, which ultimately did. To actually differentiate into that muscle cell, it had to actually create different proteins that would allow it to do the job. Okay, and so that's where we've come to. And at this point, that was very early workings of early embryonic cells. And obviously, there's more to the construction of a body and the layout of a body as the body continues to grow. And that's kind of what we're going to get into in the second half of the lecture today. In section 3 here, we're going to talk about pattern formation. Pattern formation is the development of a spatial organization of tissues and organs for these animals and plants. And basically what it allows you to do is to make sure that everything is put in the proper spot and is spaced appropriately and things go where they're supposed to go. Now, this is something that continuously occurs in plants because plants are constantly growing. Very few limitations on the size of a plant, if left unhindered. Okay, and from season to season, the plant will go dormant and then it will come back. So this pattern formation sometimes will continuously occur in plants, but oftentimes it is just limited to the embryos and in juveniles and animals. When the animal is born, chances are it's already got most or all of its pattern formation taken place. I mean, that's what you want in animals, right? You want to be able to have the animal born and everything's right where it should be. That way you've got the heart where it should be and the lungs where it should be and the arms are out here and the legs are down there, right? So in animals, pattern formation is pretty much complete by the time they're born or very, very early on in their lives. Okay. Now, the stuff that is responsible for this pattern formation is called positional information. Positional information is a generic term that kind of encompasses stuff that we've already talked about. Positional information is basically a term that means chemical messages, right? Chemical messages that we've already talked about are cytoplasmic determinants and inductive signals. That was one of the ways that we ended up the last video, talking about cytoplasmic determinants and their role in the determination and differentiation of cells, as well as inductive signals. And so these are, positional information is that generic term for molecular cues, in other words, chemical signals that control pattern formation. And it tells the cell its location relative to the body axes and relative to other neighboring cells or a particular cell in a particular organ. It kind of tells it its location relative to other organs in maybe a nearby organ system. So positional information, again, is nothing more than molecular cues, chemical messages, and you really, really do have to understand how important chemical signals and chemical information is to this entire process. It really drives all of it, and hopefully that's something that has come forward in my explanations. Okay. Now we're going to continue here with a talk of Drosophila, the development of the fruit fly over the course of its embryonic development, because a lot has been learned from a study of fruit fly pattern formation that not only applies to obviously smaller creatures, but creatures that are like us as well, a little bit bigger, a little bit more advanced. So we're gonna see the development of the fruit fly from different angles, and we're going to talk about it from different angles. First one we're here to talk about is the general life cycle of the fruit fly and kind of what happens during its formation. So we see up here in this flowchart, this is a flowchart of the entire life cycle of the fruit fly, kind of in one fell swoop. It shows you the egg at the very top, then it shows you the fertilized egg, and shows you the developing embryo, and eventually the larval and the pupae stages and the adult fly. And so what you'll notice in the egg is that the egg is very small. Okay, it's this yellow thing, and there's a bunch of other cells in the egg called nurse cells that kind of take care of it. It feeds and nourishes the egg and gives the egg what it needs. Now, after fertilization, those nurse cells are kind of done. They're gone. And we see that there is nothing here but just the egg. And this is just, at this point, this is just one cell. Okay, this is what we call the zygote, right? And after that initial fertilization, positional information is going to specify the body segments of the Drosophila. So as this fertilized egg continues to develop, it's going to have continued cell divisions, and that's what we see here. It's multinucleated at this point. And these chemical messages that are either received by the embryo or transmitted from different cells within the embryo, this positional information is going to specify these different body segments. Okay, and so eventually we get down here right before it hits the larval stage. We kind of see this embryo is segmented. And yes, it is segmented by color. The people who created the book added that. That's not actually the way it looks when it's developing, but the color is there to show you that these different body segments are different from one another. There's some segments here in orange that all kind of share some characteristics. There's some sections here in brown that share characteristics, and there's sections in green that also share characteristics. And so the positional information, these chemical signals, are going to trigger the formation of each segment's characteristic structures eventually. Okay, and so this area that's colored orange in the larvae is ultimately going to become the abdomen. It's going to create structures inside of those particular segments that would be found in the abdomen, maybe the intestinal tract or the anus or things of that nature. Okay, the stuff that's indicated here in brown is all part of the segments that are going to be turning into parts of the thorax. So, vital organs and places where the wings attach or where the wings come out of would be located up in the thorax. Okay, and this region up here in green is ultimately going to develop into the head region. It's going to develop mouth parts and eye parts and antennae parts and things like that. And so it's sequential gene expression that produces these regional differences in the formation of the segments, and ultimately when the animal turns into the adult fly, it was that positional information, those molecular cues, that told those different cells in these different segments of the fly to start developing into eyes, or start developing into wings, or start developing into legs, or whatever. Okay? So again, it is very, very important that we understand the importance of positional information and how it takes a relatively generic embryo during its development and gives it signals, gives these different cells in the embryo signals to actually turn into different types of cells based on which segment it finds itself in. Okay, so always keep in mind that it is these chemical cues that are doing a lot of this, and these chemical cues can oftentimes turn on genes that are going to code for very specific structures in these very specific regions. Okay, now I only throw this in here as a more shock value thing, and kind of make sure you understand that this information that I'm talking about here has been painstakingly discovered and observed throughout multiple generations and millions of different fruit flies. And so one of the things that they actually look at what are called embryonic lethals, which means they are flies that never actually were born. They died before they ever hatched. And so they can look at these embryonic lethals and they can identify mutations that would have affected segmentation during development. And the reason I throw this up here is to kind of give you that history behind the research, but also to shock you a little bit, to show you that the wild type that we see over on this side indicates that, yeah, it's got some eyes. This thing's like looking at you face on like I am now. You got the little mouth down here. Whoops. You got the little mouth down here, and then you've got the antennae right here. And in this mutant version, you've got the eyes, you've got right there, and then you've got the little mouth part. But now instead of antenna, you have legs. So there's obviously some kind of weird mutation here, kind of like how I discussed the other day. Maybe there was something in these grouping of cells where there's a mutation that says instead of antenna, you're going to turn on and code for legs. And so they get little legs that pop out of the antenna instead of the antenna themselves. So some weird embryonic mutations can give researchers indications as to things that might have gone wrong during segmentation formation, which we just talked about. Okay. Now, when we set up the body of the fly, one of the first things that you're going to want to do is to make sure that the fly has proper axis formation. Establishment of the axes is vitally important. You really need to know what is the front and what is the back, right? You've heard people say things like, get your head out of your butt or whatever, you know. Imagine if your head was your butt, okay? Which doesn't make sense because then it would just be your head, but whatever. It's fun to think about. But in this case, that kind of can happen, as much as odd as that sounds. And I'll show you another weird mutation that can occur here. But the establishment of the axes in fruit flies is vitally important, as well as human beings, right? We want to make sure we have a head. We want to make sure we have a posterior region. Now, there's a certain subset of genes that is responsible for proper axis formation in the fruit fly, and those gene sets are called maternal effect genes. Okay, these are a kind of a generic name for a group of genes that basically establish the axes of the body of the fruit fly. These are also known as egg polarity genes. Okay, so maternal effect genes are known as egg polarity genes because what they do is they control the orientation of the egg, and if you can control the orientation of the egg, ultimately you're going to control the orientation of the fly after it hatches, right? So that term egg polarity genes, you remember what the term polar means, right? That there's maybe a positive side and a negative side, right? The same way you can have a polarity in an egg by saying this side's going to be the head of the developing fly, whereas this side's going to be the butt of the developing fly. So the name of the gene set kind of gives you an indication as to what it does, okay? So maternal effect genes, also known as egg polarity genes, help to control the head and butt region of the fly. More specifically, even still, one specific version of the egg polarity gene is called the bicoid gene. Okay? This specific gene affects the front half of the body. It codes for a head. So if you have a properly functioning bicoid gene as a fruit fly, you should apparently, hypothetically, theoretically, have a working head region. Okay? Now, an individual with a mutant bicoid gene actually lacks the front half of its body, and it has two butts. As weird as that sounds, we're going to see a picture of that when we get to the next slide. So this phenotype suggests that the protein of the mother's bicoid gene is concentrated at the future anterior end, and even less so at the future posterior end. It turns out that the concentration of this bicoid gene's protein, once it is transcribed and translated, causes like a gradient of bicoid in the developing fruit fly, and the regions that have more concentrations of bicoid protein actually have the ability to turn into a head, whereas the areas that don't have a bunch of that bicoid gene ultimately turn into the posterior region. Okay. So yeah, we'll see a picture of that on the next slide. So this picture here shows us a wild type fruit fly larvae as well as the mutant one. And so you can kind of see here the wild type. This is where the head of the larvae is. This is actually like the mouth and then the digestive tract as it goes all the way through, and then would come out the tail region. So this is indicative of a tail or a butt or a posterior region that we see right here. And if the larvae or the embryo has some kind of mutation in that bicoid gene, the bicoid gene is not going to produce the bicoid protein properly, and it is the bicoid protein that ultimately codes for or sends a message to a particular set of cells that they should ultimately turn into parts of the head. Right? So if there is no bicoid protein, or if the bicoid protein is not working properly, then the cells in that region don't get that message, and they turn into a butt. So in this case, this mutant larvae actually has its normal tail region, but it's also got a second tail region right where its butt should be. So kind of a funny little, weird little mutation where you have a literal butt head. Okay, kind of a fun thing to do. But this is kind of how it works. So I'm gonna get to the point where I'm going to draw on this here. So bear with me as it pops up. There we go. Okay, so this diagram here illustrates what is happening with the expression of this all-important bicoid gene, as I mentioned before. Remember, I'm going to put the flow chart over here on this side. We've got things called maternal effect genes. And maternal effect genes are a larger group of genes that code for or are influential in establishing body axis. And there's a specific set of genes called egg polarity genes that do this in the egg for the head and tail regions. And then there's a specific gene called the bicoid gene that codes for the head. Okay, so that's kind of the flow of what we're talking about here, and kind of putting that vocabulary in and applying it to where we're at on this diagram. Okay, now we're going to look here first at this picture, and we notice that the egg cell, this is ultimately what's going to develop into the fly, this yellow egg cell that we see right here, and it's next to all these little nurse cells. Okay, now the nurse cells are going to turn on their bicoid genes, and when they turn on bicoid genes, they start to express those genes, right? We just established that fact here at the beginning of this chapter, that when we express a gene, we turn it on. So the bicoid genes of these nurse cells are going to turn on, and they are going to put bicoid mRNA. Okay, so when you turn a gene on and when you express a gene, the first thing that you do when you turn it on is you transcribe it. Okay, and when you transcribe it, you create mRNA. And so that's what we see here. We see this gradient, this red kind of graded coloration here. That red graded coloration tells us that there is a high concentration of bicoid mRNA in that region of the egg. And that bicoid mRNA came from these little pink kind of peach colored nurse cells out here. They help take care of the developing egg. And so we can see that in an actual microscope picture. We can see the bicoid mRNA concentrated in this region, which would basically correlate to what we see here, what I've drawn all over the red at the top. Okay, so this dark blue area is actually bicoid mRNA that has yet to be translated. Okay, so this is what happens just before fertilization. Okay, now when fertilization occurs, that's almost like a trigger. Okay, it's almost like a trigger to the bicoid mRNA that now you need to be translated, and we're going to start to make bicoid protein. So you would have like I'm going to draw a little sperm, come in here and fertilize that egg. And when that fertilizes the egg, the bicoid mRNA that we see here is going to ultimately turn into bicoid protein. And so that's what we see down here in this picture. Okay, it's a different color picture, and it looks very similar to the other one, other than the fact that this really dark orange stuff that we see here, that really dark orange stuff is actually now bicoid protein, because when we translate mRNA, it turns into a protein. And it makes sense that bicoid protein is very highly concentrated at this area of the cell, because that's where it was highly concentrated in the previous cell, the unfertilized egg, right? And so that bicoid mRNA gets translated and turned into bicoid protein in that early embryo. It's concentrated at one end, and it is this high concentration of bicoid protein that is ultimately going to tell the cells in this region that I'm kind of outlining right now, tells the cells of that region, hey, there's a lot of this bicoid protein here right now. That tells you, cell, that you are going to produce parts that would be commensurate to a head, right? You're going to produce eyes, you're going to produce a mouth, you're going to produce antennae, you're going to produce any type of hearing apparatus that the thing actually has. All of that stuff is going to be, I should say, all of those genes in those cells that are ultimately going to turn into the head, all of those genes are going to be turned on in that specific region because there's a high concentration of that bicoid that we see in that particular area. Now, one of the things you'll notice, if you can kind of just get away from all of the drawing I've done all over the thing, is that this orange coloration, as you work your way from this point to the other end, that orange coloration really does get less and less intense. And what that means is that there's actually a concentration gradient of bicoid protein. Just like any other substance that you put a drop of food coloring into a thing of water, right? It's really highly concentrated at first, and ultimately you see it kind of diffuse throughout the entire system. And that's kind of what happens here. And so this gradation, the gradient that we see of this bicoid protein throughout the course of the rest of the cell, or throughout the course of the developing embryo, really does play into which structures develop where. Okay, in areas where there's high amounts of bicoid concentration, you get head regions. In areas that's less and less bicoid, you get this lack of signal to those particular cells inherently tells those cells that they're going to develop into something else. So it's this bicoid protein that is absolutely responsible for the establishment of the head and the tail region of the fruit fly early on. And so that's a very important thing to understand. Okay, the next thing that has to occur is segmentation. So now we've set up the head and the tail region. We know what's heads and what's tails. Okay, we know what's heads and what's tails. The next thing we have to do is take a relatively generic embryo, and we have to start to give each segment, or we have to segment it up so that each one of those segments can have specific jobs, right? And so now we go into this process known as segmentation pattern. Okay, segmentation patterning, and we're going to use a set of genes called segmentation genes. They're not very creative in this case, but it makes sense, right? We call genes that are responsible for segmentation, might as well call them segmentation genes. And so these segmentation genes produce proteins that produce the formation of segments after the axes are formed. Now, there's different types of genes. Okay, positional information is provided by sequential activation of 3 different sets of segmentation genes. So the segmentation genes themselves is kind of this overarching generic group of genes, and within that grouping there are 3 specific segmentation genes that are going to be activated. They're called the gap genes, the pair rule genes, and the segment polarity genes. Okay, now I like this flow chart because it kind of puts everything into perspective here. One of the first things that needs to happen when we are trying to build a body is to make sure that we know what's heads and tails, what's top and bottom, and that's what the maternal effect genes do. And specifically the one we talked about, the egg polarity genes, set up the head and the tail regions of the fly. Now, once the axes are established, these segmentation genes are turned on, okay, and they are turned on in sequence. The first ones to turn on are called the gap genes. Once they do their thing, they turn on the pair rule genes, okay, and then we turn on the segment polarity genes. And each one of these gene sets kind of does something specific, right? So the gap genes are going to take the segments of the fly, which were starting to be set up by the maternal effect genes. It's going to take these genes, or pardon me, these segments of the body, and it's going to make sure that they're properly spaced. That's what the gap genes do. Okay, make sure that the abdomen has this much space, and the thorax has this much space, and the head has this much space. Okay, that's kind of what they do. Provides positional information. Each one of these genes do. Pardon me. Provide positional information to these cells of these specific regions of the developing fruit fly, and says you get this much space, and you get this much space, and you get this much space. It's providing proper gaps, right? Providing proper gaps in between the different regions so that they have enough space. Okay. The pair rule genes kind of do exactly what they say. They look at each segment of the developing body and say, okay, here in the thorax, we're going to make sure that each segment has, each segment has 2 legs. Okay, there's 2 legs there. Now we're going to have, okay, there's 2 legs here, and then there's 2 legs here. Okay, so everything that should have a pair has a pair. That's what the pair rule genes do. And the segment polarity genes, so even within the segments, you got a segment here, you want to make sure that you know that this segment is closer to the head and this segment's closer to the butt. Okay, so there are certain sets of genes that will also allow for polarity to be identified even within each identifiable segment of the animal. Okay, so it makes sense. A larger animal or a larger polarity is established. So this is the head of the animal, this is the butt. But even within then, you want to make sure that those segments know where they're at in position to everything else as well. That's why that term positional information is so important, because it does provide information about the position of these different parts of the animal's body as it gets bigger. Okay. The next set of genes to be activated are called homeotic genes, and those are the ones we're going to talk about next here. Homeotic genes are responsible for the anatomical identity of each segment. Okay, to say, okay, when these homeotic genes turn on here, because of this specific piece of chemical information, that means that the head region is going to form. So eyes are going to form here, and antennae are going to form in this specific spot, and the mouth gets put in this specific spot. Okay, and then we look at the thorax. All right, in the thorax we've got legs, and we've got legs, and we've got wings. All right, we want to make sure that the leg genes are turned on. Okay, we're going to use some homeotic genes to make sure that the legs get turned on in a very specific spot, and then the wings get turned on in a very specific spot up here too. Okay, so here in the abdomen we've got other things here in the abdomen, like the anus and other digestive, other types of digestive, structures, things of that nature. So different cell groupings in those different parts of the abdomen are going to be turned on through homeotic genes and given an identity. Okay, certain genes are going to be turned on that say you're going to be part of the intestine system, you're going to be part of the anus and how that works and everything else. So the anatomical identity of that fruit fly segment is set by these what are called master regulatory genes called homeotic genes. And so this is oftentimes where we see the kind of crazy stuff that I've showed you up until now. We see the leg coming out of where the antennas should be. We see the eyeball coming out at the very tip of the antenna. Chances are there's some kind of mutation in a homeotic gene that says, all right, this mutation says you're going to get a leg here instead of an antenna. Okay, and so that's what we see actually in this picture. Again, very similar picture to the one I've already shown you. Same thing we see at the very beginning of this lecture, where you got the little eyeball coming out of the tip of the antenna. Chances are at the tip of the antenna in some part of early development there was mutation in the homeotic genes that basically said instead of completing this antenna, you're going to create an eye. And so the homeotic genes told it to create an eye because of that mutation. So kind of a funky, weird thing, but when you're using fruit flies and you can get hundreds of thousands of millions of offspring, you're going to see weird stuff like that sometimes. Okay, so let's talk a little bit about the nematode. Okay, we're going to kind of get off of fruit flies, kind of learn that's a very basic, scratch the surface discussion of how they set up their body plans. We're going to talk about another famous research creature called C. elegans, which is known as the nematode. And what's interesting about the nematode is that they know it's a very simple creature, and it's cool because you can see through it. It's translucent. You can look at every single thing, every single structure inside of it because you can see through it. And so what that allowed researchers to do is to basically track every single cell division from the very first. Okay, so they can watch the zygote as it divides throughout its full development, and it can see every single cell division and know where everything happens. And so they know almost every single thing they can about the development of these different structures, these different cell types, which is ultimately what we're talking about, right? These different cell types in the nematode that you see below, okay? And so one of the things I want to discuss, and this is one of the questions you've already seen, and I wouldn't be surprised if you see it again on this test because I like it. It's applicable in the cell signaling chapter 11 test, and it's also very applicable here. And I think I had alluded to that before. So in the development of our friend nematode up here, very early on, researchers figured out that even at the 4-cell stage, there's a very important piece of positional information that is shared between 2 cells that will ultimately affect the developmental fate of different organ systems within the nematode. Okay? And so the cell signaling of one cell to the other, known as induction, which we've kind of talked about already, is absolutely important to the proper development of this organism, and in some cases us as well. If we don't get certain signals sent at certain times during our development, we're born with what doctors call birth defects. Okay, so I've talked about this before when we discussed it in the Chapter 11 assessment, but the interaction happens between cells 4 and cell 3, cells 4 and 3. Okay. We can see a zoomed-in version of what's happening here. Cell 4 is the one responsible for providing some kind of inductive signal to cell 3. And what this inductive signal tells cell 3, it provides a message that says, okay, cell 3, next time you divide, your posterior daughter cell is ultimately going to form all of the intestines for the creature. Okay? And your anterior daughter cell, the one that's in quote-unquote in front, will go on to form muscles and gonads of the adult creature. Okay? And so one of the things that they can do is they can mess with nature a little bit, and they can remove cell 4 right before this signal is sent, researchers can, and they can witness what happens to the developmental fate of the daughter cells of cell 3. Okay. Upon the next division, when there's no signal, the nematode doesn't get intestines. That's not very nice of it, but that's what happens. If you don't get the signal, you don't get intestines. Okay, so that signal has to be given to cell 3 upon its next division. It tells it exactly what it's got to do. Okay, that this half, okay, the right half of the daughter cells of cell 3 is going to turn into the intestines, and the left half would turn into the muscles and gonads. So it is that signal that's vitally important. So this is just kind of a list of the different things that can be gleaned from a study of the nematode. But I just wanted to relate that because that's a real-world example of something we talked about in the very first video when we discussed induction. Okay. There's also a phenomenon I'd like to talk about called apoptosis, and I believe that we've mentioned this before, at least in some context. And apoptosis is vital to the normal creation form of these organisms because it allows certain things to be destroyed. Okay? Apoptosis is programmed cell death, and programmed cell death ultimately leads to the formation of, in some cases, digits. Okay, so maybe early on in our development, we did not have discernible digits like we do now, 5 fingers or 4 fingers and a thumb. It would have looked more like a webbed mitt, almost kind of like a baseball mitt. And there are some, and I think I've drawn on a picture at some point like this before. Might as well do it again since I'm making a video. Bugger. All right, hold on. Bear with me. Okay. So all of this is cellular tissue, right? And you can kind of see the shadow of digits here. Okay, I'm going to outline them. These are going to be what ultimately become the digits. They're a little bit darker than some of the other ones, okay? And in between these digits, you see extracellular tissue. Okay, that we see right there. And obviously we don't see that right now, right? It's gone. And so at some point that tissue had to have been quote-unquote eaten away or would have had to have died. And we kind of see that happening here. We see the digits a little bit more discernible now. Yet we still see some tissue in between. So that tissue is going to be killed. It's not needed. It's waste tissue. And the body will reabsorb those cells and any nutrients that they might be able to give. And ultimately, by the time this apoptosis happens with the webbing, with the cells of the webbing of the fingers, we get some discernible digits. And this happens as well with most of us. Humans do at some point in their development have what's known as a vestigial tail. Okay, and most oftentimes during our development, the tail tissue undergoes apoptosis, and 99.9% of us are born without tails. But there's still always that little blip in the system, if you will, where apoptosis doesn't work properly in some individuals at that particular stage of development, and they're born with little tails. There are pictures I've got in another PowerPoint where we'll discuss vestigial structures and what they can kind of maybe indicate to us about evolution, but I'll wait and leave those pictures for that particular lecture. If you want to go ahead and look those up, feel free to look up pictures of vestigial tails. Some of them are a little bit shocking, some of them are Photoshopped, some of them are real. But it's always kind of fun to look at that. But that's just another example of a structure that we no longer have, when we're born, that was eaten away by apoptosis. So even something as odd as it might sound, something like programmed cell death or the death of cells can be responsible for the formation of our body and what we look like after we've been totally created. So it's a really, really interesting process, all of this. And like I said, we've only just barely touched the surface of it. There's so much more that goes into it. There's so many more gene sets. There's so many more body structures and body systems that need to be discussed in detail. But at least now you have kind of a big picture idea. You've got this idea of the fact that chemical signals and sequential gene activations that have to happen in a certain order are really, really vitally important to how we develop as growing embryos and ultimately adults. So I hope you enjoyed that. I know I always do. I don't understand a lot of the intricacies of this system. It gets far more difficult than that. But it always just amazes me, and I always just sit and think about what I do know and how much then, again, that I don't know. And, I think I mentioned it in class at some point, but there's trillions of events that all have to happen in just the right order and in just the right way for us to be where we're at. And so it's a minor, I would say major miracle that any of us have ever developed to the point that we are. But do try to keep that in perspective and try to understand that we have really only barely scraped the surface of everything that goes into this. So I hope you enjoyed it. I know I did, and I'll talk to you soon.